Hello, and welcome to this Nature.com custom webcast titled Basic Principles of Clearing and Imaging Biological Tissues. My name is Joshua Carpen, and I will be your moderator. Today's webcast is sponsored by Logos Biosystems. We'll begin the webcast with a presentation from Dr. Doug Richardson of Harvard University, and then end with a Q&A session. To ask a question, just type it in where it says type your question here, and then press submit at any point during the webcast, and we will answer them today. And now over to Doug.
Hello, and welcome to today's webcast on the basic principles of clearing and imaging biological tissues. My name is Doug Richardson, and it's a pleasure to be with you today. I'd like to begin by saying thank you to our sponsor, Logos Biosystems and Nature.com, for the invitation to present to you some of the work that we've been doing in the Harvard Center for Biological Imaging in regards to tissue clearing. Just before we get started, a really quick disclaimer: I'm not receiving any financial compensation for this webcast today, and Harvard University does not endorse any of the commercial technologies that I will speak to you or present about today. It's just a quick outline of what I'd like to cover with you today. First of all, I'd like to talk about the physics of light scatter in biological tissues. After we've got an idea of what happens when light comes in contact with tissue and why that can be a problem when we're trying to image under the microscope, I'd like to move on to the basics of tissue clearing. So basically the molecular mechanisms behind the tissue clearing methods. And today we're going to be focusing on the clarity technique. After that, I'd like to move on to spend a little bit of time talking about how we image cleared tissues. So what are our options on the microscopy side, and what are the considerations that we have to keep in mind when we're placing a piece of clear tissue under our microscope. And then finally, I'll just close by giving a couple examples from some work we've done in our facility and give you a quick rundown of some of the tissues that we've worked with through this process. So I'd first like to introduce the idea of light scatter with this very simple experiment. What we have here are two plastic cubes. Both of them are filled with water. The cube on the left just has pure water in it, whereas the cube on the right has pure water, and I've also dropped 10 microliters of whole milk into there. If you hold these two cubes up side by side, you cannot tell the difference between them. They look absolutely identical. But you can see here when we take a laser pointer and we shine that laser light through both cubes, there's a very obvious difference. So on the cube on the left, that light enters the cube, goes directly through the cube, and exits on the right-hand side. And we would generally describe this cube as having no light scatter in it, meaning that that laser light is going directly from the left side to the right side, and none of that laser light is scattering off the water and coming towards the camera, or in any other direction other than the forward direction of the laser light. Whereas in the cube on the right-hand side, the laser beam, as it passes through that cube, is clearly visible to the camera. And so this means that as that laser is traveling through that cube, it's coming in contact with something that is scattering it, and it's scattering that light out towards our camera. In reality, it's scattering it in all directions. No matter which angle you look at it, you're going to see that laser line going through that cube. So what this means is, in the cube on the left, the intensity of the laser beam that enters the cube is approximately equivalent to the intensity of the laser beam that comes out of the cube. There isn't a lot of laser light loss there, or laser intensity loss there. In contrast, in the cube on the right, the laser light that enters the cube is more intense than what comes out on the right-hand side of the cube, as a lot of that laser light, or some of that laser light, is being lost to this scattering effect as it passes through. And this is essentially what's happening as we use a laser beam, let's say, to excite fluorophores in biological tissue. The deeper we go into that tissue, the less laser intensity there is. And the exact same thing is happening with our fluorescence on the way back out. So if we excite a fluorescent molecule deep in a piece of tissue, that fluorescence isn't able to get back out of that tissue to our objective. So next, let's take a look at a real biological example. So here we have a couple of pieces of brain tissue sitting inside a petri dish. The first thing you can notice is that these tissues appear white. So what that means is that all wavelengths of the light that's trying to pass through that tissue is being scattered equivalently. So the blue, red, and green light is all being scattered. And what this is going to mean is that it's going to be very difficult for us to image deep into this tissue. So we have a limit on our imaging depth that's imposed by this natural scatter of the biological tissue. And so in general, with a one-photon excitation process using a traditional confocal microscope, you're going to be limited to pretty much a maximum of 200 microns. And that is really a best-case scenario. In many different tissues, and depending on your labeling strategy, you might even only get 50 microns into the tissue before your image quality degrades drastically. So the invention of two-photon microscopy allowed us to increase this imaging depth. So by using infrared light, which penetrates deeper into biological tissue, and by only exciting fluorophores at our focal plane, which means that we can be more efficient in our collection of the emission photons coming out of that tissue. We can just collect everything. We don't need a pinhole anymore. We're able to image deeper into the tissue. So with the two-photon excitation process, you can get close to a millimeter into the tissue. Again, this is a best-case scenario. Not every tissue, not every labeling strategy is going to permit that. So what does this look like under the microscope? So here's an example of Eric Fetzig's group, and this is imaging into the brain of an intact zebrafish, which is one of the most optically clear tissues that we routinely image in biology. But I think you can still appreciate here that as we move deeper and deeper into the fish, our image quality is suffering. So by the time we get down to 200 microns, our image has a lot of what looks like sort of background fluorescence or non-specific fluorescence, which is actually a lot of the scattered light. And the detail of, or the contrast on our smaller objects, like the axons and dendrites that are labeled there, are very difficult to see. So this is commonly what happens when you start imaging deep into biological tissue. So the Fetzig group, they're actually able to clear this up using a technology known as adaptive optics and some deconvolution. So you can see even at 200 microns now, they're getting nice clear images. These technologies are a little beyond what I'm going to talk to you about today, but I just want to point out that as fantastic as these technologies are, there is still going to be a limit here. This is going to allow you to go a little bit deeper, but by no means this is going to allow you to image to the depth that you can by doing a tissue clearing process on that tissue ahead of time. So let's return to that initial experiment that I showed you, where we have the two cubes of water, the one on the left was pure water, the one on the right that has a little bit of milk in it, and our laser beam passing through both of them. Let's take a look at just the cube with the pure water, the one on the left first, and ask what's actually happening inside that cube as the laser light comes into it. So if you take a look at these little diagrams on the left here, let's look at panel A first. And the horizontal arrows there represent the laser light coming into the cube. And these colored stars represent the first row of water molecules that are just inside that cube. What's going to happen is the laser light's going to come in contact with these water molecules, and some of that laser light is going to be scattered by the water molecules. So what does that actually mean? What it means is there's a photon of that laser light that hits the water molecule. For a very brief moment, that photon gets, the energy from that photon is absorbed by the electrons around that water molecule. They vibrate a little bit quicker. And then another photon is eventually released from that water molecule. It looks absolutely identical to the one that came in. The only difference is the direction which it is now propagating in. And that can be in any direction. It has no relation to the initial direction of the laser light coming into it. So it can go left, right, up, down, in or out. It doesn't matter. So now if we move over to panel B, what we can see is there's a really interesting relationship between the scattered light that comes off of each one of these little colored stars that represent the water molecules. And this has to do with the way the water molecules are packed in there. They're all packed in there obviously nice and tight to one another. And what that means is that whenever light is scattering from one water molecule, there's always going to be another water molecule about half a wavelength away from it that's also scattering light. So if we take a look at the blue star at the very bottom there, we can see the black line coming into it represents our wave of laser light coming in. It contacts that blue star. Some of it is scattered, and we've illustrated the scatter here as being in the upward direction. So we can see the blue wavy line coming off the star is the scattered light. Identically, the yellow star, the yellow water molecule, is also scattering light, and that's this yellow wavy line. If we look at those two wavy lines, they're both perfectly out of phase with one another. And what this means is that we're going to get destructive interference when these two waves interact with one another. And exactly the same thing with the green and the red star. They're going to be destructively interfering with one another as well. So what this means is that any direction other than the forward direction, this process is going to happen. So there's always going to be another water molecule that's also scattering light that's perfectly out of phase. So it doesn't matter whether this is up, down, reverse, into the plane of the paper, out of the plane of the paper. Every single direction except the forward direction, there's going to be this destructive interference of that scattered light. So now if we move down to panel C and we look a little bit differently, let's take a look at molecules that are lined up horizontally in the same plane as that laser light that's coming in, and let's see what happens here. So similarly to the diagram above, that laser light's going to come in, but in this instance it's going to scatter sequentially. So it's going to scatter from the red, and then from the blue, and then from the green, and then from the yellow. So if we look at panel D, what we see here is our black line representing our wave of laser light coming in. And at the very top, at 1 femtosecond there, it contacts that red molecule of water. And it's going to scatter. So what that means is there's going to be that brief pause where it interacts with the water molecule, the photon of light interacts, and then it's going to be released at 2 femtoseconds. So if you go down to the 2 femtosecond line, you can see the red wavy line beginning there. But at that point, that initial laser light is scattering off the blue molecule. And so if we go down to 3 femtoseconds, we now see that the scattered light from the red molecule and the scattered light from the blue molecule is actually constructively interfering. So these are actually adding up with one another. And this continues with the green molecule and the yellow molecule. So only in that forward direction are we actually getting an additive effect of the light. So what this means is that generally we would describe this left cube as having no light scatter at all. And that's not true at all. There's lots of light scatter happening in the left cube. The difference here is that light scatter is only constructive in one direction, in the forward direction. So that's the only spot where all that light is going to go. Any of the light that's trying to come out towards our camera is destructively interfered, and we don't see any of it in that direction. Everything is moving forward. So let's take a look at the cube on the right-hand side here, the one where we described it as having lots of light scatter because we could see the laser beam as it passed through the cube. We still have a lot of water molecules in here that are packed in pretty tight, but we've also added that milk into it. And one component of milk is large lipid droplets. And so in the diagram on the left here, I've depicted our water molecules as blue circles and our lipid droplets as red ovals. And so what's happening now is those lipid droplets are spreading apart the water molecules. So they're more than a wavelength or half a wavelength away from one another. So they're no longer able to find a partner that's going to deconstructively interfere the light that they scatter. Similarly with the lipid molecules as well, they're going to scatter some light, but they're not going to have another molecule exactly half a wavelength away from themselves. So because of this, we're now getting this light, our scattered light, the same as in the initial cube. We're still getting lots of light scatter, but now we no longer have that destructive interference in all of those other directions. So in this case, we just have light scattering in all directions with no destructive interference whatsoever. Now, obviously, this doesn't happen every time you mix two substances together, so it's possible to mix two substances together and still get a laser beam passing through the cube just exactly like it does on the left-hand side. And so the question is how do we measure whether or not two substances are going to create this destructive or constructive interference in the lateral directions? And the easiest way to do this is to look at the refractive index of the two different substances. If those two substances have vastly different refractive indices, we are going to lose that destructive interference, and we're going to have this situation that we have in this cube on the right, or in biological tissue where we have lots of light scatter. So what are the different refractive indices that exist in tissue that give rise to this cloudy mess that we're trying to image through? Well, there's a couple components that are really key to this. So obviously we have a lot of intracellular fluid, extracellular fluid that is primarily made up of water and salts and a bit of protein. This usually has a refractive index around 1.35, so just a little bit higher than water. One of the biggest scattering centers, what contributes the most to this cloudiness of biological tissue, is lipids. And in general, this isn't the plasma membrane. The plasma membrane is quite thin, and so there isn't a lot of time for it to interact with light as light passes through it. But areas like mitochondria and Golgi, where we have double membranes that are folded in on top of each other, these are really the areas that create an area of high refractive index within a cell. So these can be up in the range of 1.45, 1.47, quite a bit different from that intracellular and extracellular fluid. Proteins as well can contribute to this. So especially if you have hydrophobic regions within a protein, but once again, these are generally quite small in relation to the wavelength of light and the amount of time that light interacts with them. But structural proteins like collagen, actin, myosin, that are very repetitive, these often have very high refractive indices, and these can be in the ranges of over 1.5. So these are three of the key refractive indices that we have within a cell. And so if we really want to do a tissue clearing process and turn that tissue as clear as we possibly can, we need a way to make these all equivalent all the way through that tissue. So the real true key to tissue clearing is matching that refractive index all the way through your sample so that we get that nice homogeneous interference, that deconstructive interference in all directions except the forward direction, where we have some nice constructive interference as our light goes in and exits the tissue. And this is what's going to result in a nice clear tissue. Now, tissue clearing has been around for over 100 years, but we've really seen an explosion of methods in this area in the last decade, 15 years. There's just been a tremendous explosion in the amount of named techniques that are out there. And it can be a little confusing to someone when you first sit down and try to select one of these. A couple of years ago, Jeff Lichtman and I tried to categorize these to make it a little more simple. So we came up with these various categories of solvent-based clearing techniques, simple immersion techniques, hyperhydration techniques, hydrogel embedding techniques, and then hydrogel embedding plus hyperhydration. So for the talk today, I'm just going to be concentrating specifically on one of the hydrogel embedding techniques, the clarity technique. But just to let you know, there are all kinds of these out there, and it may be worth exploring those depending on your particular sample. No matter which tissue clearing method you choose, there are a number of steps that are common to each one. I've broken it out into 4 different steps here. Now, sometimes these are combined into a single step in various methods, but underneath they all do these 4 basic principles. So first, there's often some sort of pre-treatment to get your sample ready for the tissue clearing process. Then there's a permeabilization or lipid removal step. You may do some immunolabeling if you don't have fluorescent proteins in your sample, or if your particular method quenches the emission from those fluorescent proteins. And then you're going to move on to the final clearing refractive index matching step. So it's really important to remember that your sample is not going to go clear until you get to that final clearing step. So there's going to be a lot of steps in between that you're going to do to process your sample. It's still going to look cloudy, and it's not going to get clear until you get to the final, very end there. So today we're going to be focusing on the clarity technique. So I just want to show you a couple steps specific to the clarity method. So in the pre-treatment, depending on what type of sample you have, you may need to do some bleaching ahead of time. So if you have a sample that has a lot of pigment to it, you may need to bleach that out. You may have some autofluorescence that you also need to try and treat. But the most important step in the pre-treatment of the clarity procedure is this hydrogel embedding. We're going to talk about that in some future slides. The next step is going to be to remove the lipid as best as possible from that sample. We're going to do this using a detergent SDS as well as electrophoresis. Once you've got that lipid out of there, it's a little bit easier to do your immunolabeling at this point. It's easier for the antibodies to diffuse through your sample. But there's certainly some antibodies out there that are happier or do a better job of finding their epitope if you do this at the very beginning of the process. So this immunolabeling can be done now, it can be done at the beginning, or of course you obviously don't have to do it at all if you have some fluorescent proteins in your sample. From there we're going to move on to our final clearing and refractive index matching step. And there's a number of solutions that have been used for this. The one commonality between them is they mostly are all based on a high refractive index medical GI imaging contrast reagent. So there's a number of commercially available ones. Focus Clear, the Logos Biosystems mounting medium are two of those. There's also some homemade options, RIMS, Protos, OptiView that you can choose from. Now let's take a closer look at each one of these steps. So first, the hydrogel embedding step. Now most of you are probably starting with an intact animal. And the most important thing here is going to be to do a really good perfusion with saline. So you want to use a large volume of saline and get as much blood as you possibly can out of that animal. Any blood that remains is going to give you a lot of autofluorescence in the vasculature. So keep a close eye on the organs. Make sure things like the liver and the kidneys are really turning nice and pale yellow or white, and make sure you're getting as much of that blood out as possible. Next, you can either perfuse with paraformaldehyde to do a fixation, or you can even perfuse with the hydrogel solution at this point. Personally, I prefer to perfuse with paraformaldehyde. Then you can take your tissue of interest out of the sample. You can do some bleaching or autofluorescence reduction if you need to at that point. And then you can sit that tissue in the hydrogel solution overnight in a fridge. And this allows that hydrogel solution to penetrate in nice and deep. So what's the hydrogel solution made up of? Again, I said we're going to sit this in the fridge overnight, or for about 24 hours. And it's made up of paraformaldehyde, acrylamide. Some people put bisacrylamide in there, and this chemical called VA044, which is a temperature-sensitive crosslinker. This is exactly the same as, or serves the same purpose as TMED in an SDS page gel. It cross-links the acrylamide, but it will only catalyze that reaction when the temperature of the sample has been raised. So by this way, you can control when that actual polymerization of the gel occurs. There are now commercial options to help you with the polymerization of this hydrogel solution. So once you've let your sample sit in it overnight or for about 24 hours at 4 degrees, what you're going to want to do is warm that sample up to cause that temperature sensitive crosslinker to do its job. Now, it's pretty important that you do this in an oxygen-free environment. Oxygen is going to inhibit the cross-linking of that acrylamide. You're not going to get as substantial a hydrogel throughout your sample. So it's really good to do this in an oxygen-free environment. And it takes about 3 hours at 37 degrees. So this is an example of one of Logos's, their polymerization system for doing this. You essentially have your sample in a Falcon tube or a multi-well plate. And this system, it's just a vacuum chamber that removes all of the oxygen so that you don't have to worry about that. And then it heats things up to 37 degrees. Now, in some situations, it may be advantageous to play around a little bit with the hydrogel matrix. So this is an example from the Garadnaru lab at Caltech. And they gave an example of what can happen when you play around with the relative concentrations of acrylamide and paraformaldehyde. So on the panel on the furthest right here, where it says A4P4, this is the composition of hydrogel that was originally published in the first Clarity paper out of the Diceroff lab. And A4P4 stands for 4% acrylamide, 4% paraformaldehyde. You can see you get a nice tight gel with very small pore size in it. Now, in this particular paper, the authors also showed two other compositions, so A2P0 and A4P0. So that's 2% acrylamide, no paraformaldehyde, as well as 4% acrylamide, 0% paraformaldehyde. And what they found here was that they found the A4PO composition, A4P0 composition, was the most efficient at allowing lipid to be removed as well as immunolabeling to occur. So this larger pore size just makes it easier to get things in and out of your tissue. Probably has a bit of a disadvantage that it's also going to preserve a little bit less protein in there, so you're going to lose some protein as well. But this is one option that you have, is playing around with this pore size. So if you want to increase this pore size, there's a couple options. One is to use less or no bisacrylamide, as was shown here. You can use little to no paraformaldehyde. You can decrease your acrylamide. And the other thing you can play around with is the amount of cross-linking time. So on the last slide, I told you about 3 hours. If you shorten that time, you're obviously going to get less cross-linking as well and larger pore sizes. Okay, so now you've got your tissue nicely embedded in that hydrogel. You've set it in PBS overnight to allow any paraformaldehyde or acrylamide monomers that didn't cross-link to diffuse back out. And now you're ready to remove your lipid. So this is done in a solution made up of SDS and boric acid. And depending on the size of your sample, this is generally done for 5 to 24 hours, but it may even need more time than that if you have something that's really enriched in a lot of lipid, or if you need something that needs a slightly lower current while you're doing that clearing step. So again, this is Logos product for doing this. So you can see there's a power control unit on the right there, and on the left there's a little chamber. That's where you're going to put your sample inside of it. It's an electrophoresis chamber. We're going to flow that SDS boric acid solution through that chamber from the reservoir, the big plastic bottle out the back there. And that system is just going to sit there for however many hours we tell it and slowly start to remove all of this lipid from our sample. So our sample has now come out of the electrophoresis chamber. We're going to put it in PBS overnight again, just to allow all of the SDS that's remaining inside of it to move out of the tissue. And then we're all ready to go into our final refractive index matching or clearing step. We're going to use one of those clearing solutions that I spoke to you earlier, one of the commercial ones or a homemade one. And what's really important here is the volume of those solutions that you use. So these solutions are unfortunately pretty expensive, so most people try to use as little as possible, and that ends up being a large mistake. So if you take, let's say, for example, an entire mouse brain, and you put it just in 3 mils of the solution, just so it's just covering it, you probably have at least that amount of volume of PBS inside your brain. Those two solutions are going to equilibrate with each other over time. You no longer have a clearing solution, you have maybe 50% clearing solution, 50% PBS mix. Your tissue will not go clear. So you cannot be stingy with the volumes of these solutions that you use. For an entire mouse brain, what I do is 20 to 25 milliliters for 24 hours, and then I take that brain and I put it in a newer, fresh solution for another 24 hours, and that one usually is 30 to 35 milliliters. The one thing that is nice about these solutions is you can reuse them to some extent, so that can save you a little bit of money on that side. So large volumes is very important. Also, whichever solution that you use for the second round, that should be the exact same solution that you keep that sample in during imaging. Don't then put it in another fresh round of solution. You're going to have some refractive index mismatches there that interfere with your imaging. Okay, so we've got 3 brains here. These are 3 separate samples. The occipital bulbs didn't just magically appear. But you can see the brain on the left is what it looks like after the hydrogel embedding step. The one in the middle is come out of the electrophoresis chamber. It's normal to see a little bit of expansion there. You want to try and keep this as minimal as possible. And then once it goes back in the final refractive index matching solution, it should shrink down a little bit again. Great. So we've gone through the clearing process. We now have a nice cleared piece of tissue. And the next question is, how are we going to take a picture of this? So obviously we're going to turn to one of our nice modern day imaging systems like this one here. Of course I'm kidding. One of the big difficulties though is every microscope has always been designed very similar to this one. I mean, this isn't that much different than the microscope that you're going to buy today from a manufacturer. And these have all been designed with a particular sample in mind, just like the ones that are scattered all over here. So we're always thinking of a microscopic sample as being a nice little thin section sitting in between a cover slip and a glass slide. And that's because this is the way we've done it for years and years and years. And it hasn't been until recently that we had the ability to start looking in 3D. But as I showed you on the first slide, even with confocal microscopes, in the past we've only been going usually a few hundred microns at most into that tissue. So a microscope never had to be designed that was able to necessarily fit a sample that's a couple millimeters, a centimeter thick inside of it. So this is one of the biggest struggles that we run into when we're imaging cleared tissue, is finding a microscope system or design that's able to accommodate the samples that we have. And then the second problem we have is on the back end, a lot of data handling issues. So if we go into a little bit more detail here, these are what I find to be the 5 biggest barriers to imaging cleared tissue. The first is the working distance of the objective. If your working distance of the objective is smaller than the distance you want to image over, at some point that objective is going to run into your tissue and you're not going to be able to image any deeper. The second is these clearing solutions that we're working with are often made from refractive indices that don't match the design of the objectives in our lab. So they don't perfectly match the refractive index of glycerol or water or oil. Thirdly, some of them are made up of solvents. And this generally isn't a problem with the clarity technique, but sometimes we have to watch out for solvents that can damage our microscopes. Finally, we have to deal with the amount of time it's going to take to image a sample. And lastly, as I mentioned earlier, we're now going to have a lot of information from our sample, and we're going to have to figure out some way to store this data and some way to analyze it and visualize it. So the good news is a number of manufacturers have now addressed a lot of these concerns and created a number of objectives that are designed specifically for tissue clarity imaging. So in general, these are all low magnification objectives, which is nice. They give you a large field of view, allow you to image a big piece of your tissue all at once. So in general, less than 25x magnification. They all have very high numerical apertures. So remember, it's not the magnification, but the numerical aperture that's going to determine the resolution of your image. So these all have NAs above 0.9. Probably most importantly, they have a really long working distance. So they can all image more than 5 millimeters into tissue. They're dipping objectives, which in general makes it a lot easier to mount your sample. You don't have to worry about creating some sort of chamber with a cover slip or anything like that. But just be careful, not all of them are to be used with solvents. Some of them definitely can be, which is great, but not all of them can. And they are all refractive index matched to the different clearing solutions. So some of them can handle a wide variety of clearing solutions with nice little correction collars on the sides of them, and others are designed for a more narrow purpose. Unfortunately, not everyone has access to these really high-end tissue clearing specific objectives. So I just want to point out 2 common mistakes that I often see people doing when imaging clear tissue, and a couple solutions to avoid this. So the first one that I often see is people will try to substitute a water dipping objective. And you can see underneath there, this satisfies most of those important criteria that we have. Unfortunately, this objective is designed to dip into a solution with a refractive index of 1.33. Our clearing solutions don't have that. They have a higher refractive index. So if we do a little experiment where we take a petri dish, put some 100 nanometer fluorescent beads on the bottom of that dish and just cover them with a little piece of agarose, and we fill that dish with water, and let's just look at what those beads look like. So in the 4 panels on the right-hand side here, the 2 panels on the left, these show a point spread function of one of those beads. So this is just a single bead imaged on a confocal microscope. And what you can see is when that dish is filled with PBS, which has a refractive index which matches what this objective was designed for, we get a nice tight XY point spread function, and our XZ point spread function looks pretty good as well. But if we replace that PBS with glycerol, so these are now the 2 panels on the far right-hand side, you can see our XY point spread function. It's not too bad, but it's definitely gotten worse. And where we really notice this is in the axial dimension. So our point spread function in XZ is just very blurred, very drawn out. And what this means is that when you're trying to do 3D imaging, that image is going to look very poor. And this just has to do with the large amount of spherical aberration that's being created in this optical system. So this is definitely something you want to avoid. The one way to get around this is that spherical aberration is going to be proportional to the numerical aperture of the objective. So this is one of the rare cases in imaging where going to an objective with a lower numerical aperture is actually going to give you a better image. So if you go down to a 10x water dipping objective with say like a 0.4 numerical aperture, even 0.5, you're actually going to get a much better image. And that can be a good substitute for some of these more expensive objectives if you don't have access to them. Another option is to use a long working distance air objective. So for example, this 10x objective that we have here. Again, there is something that can go wrong here. So if you look at this sort of complicated schematic, what I've put in the top right here, if we look at the objective on the left-hand side, the one that says it's in position 1, I've diagrammed the peripheral rays that are coming out of that objective according to a numerical aperture of 0.8. You can see there's some black lines and some blue lines there. The black lines represent where the focal point of that objective would be if the refractive index was identical on both sides of the cover slip. Okay? So kind of where you expect the focal point to be of that objective. But of course, if we have a clearing solution there, let's say a high refractive index clearing solution, and we are using an air objective, there's going to be a refraction event at that surface. And so the blue lines actually represent where our focal plane ends up after that refraction event happens. Okay? So what we're going to do now is we're going to simulate doing a z-stack through that sample. We're going to move that objective down closer to our sample to position 2, which is depicted on the right. And so the red arrow in between the 2 objectives, right above where it says cover slip, this shows the distance that that objective has moved. Again, with this objective in position 2, we've drawn the black lines simulating what would happen if the refractive index was identical on either side of the cover slip, and the blue lines that simulate what happens when we have a higher refractive index below the cover slip. And so what you can see is if we compare that red arrow that I was talking about earlier, which is the distance that the objective physically moved, and we look at the orange arrow at the bottom there, which is the distance that our image plane actually moved, you can see that orange arrow is much larger. So the microscope software thinks that we've moved the distance of the red arrow, but we've really moved the distance of the orange arrow. So what this means is when we actually take a number of z-slices through that sample and we try to put those back into a 3D volume, the microscope thinks that we've moved less distance than we actually have. So if you look at the images of the spheroids at the bottom, let's look at the box on the left-hand side where it says uncorrected. This is an image of a spheroid, a 3D tissue culture ball of cells that has been grown. It should be roughly spherical. And in the XY dimension, it does look that way. But when we do a projection of all of our Z slices in the YZ dimension, we see it looks kind of squished, a little bit like a pancake or an oval. So what we need to do is we actually need to correct this image. We need to tell our software that, hey, we actually moved further in the sample optically than the physical movement of the objective. And this is what's been done on the panel on the right. So once you apply the proper correction to this, you can see we now have a nice sphere in the YZ dimension as well as in XY. So our next barrier is just the sheer amount of time that it's going to take to image a sample. So I sat down and did a calculation a while ago. If you wanted to image an entire mouse brain that's been cleared with a laser scanning confocal or 2-photon microscope, you had an objective that had a 20x magnification, you did a 1 micron Z-step, and you were able to image one Z-slice at a time. You actually need over 4.2 million images to do this, and it's going to take you about 50 days. So what this really says is with a confocal microscope, you're probably going to make, this is a point scanning confocal, not a spinning disk, you're probably going to make some compensation somewhere that's going to reduce your image quality, it's going to reduce the resolution. You're not going to get what the actual achievable resolution out of the system because you're not going to wait 50 days to do this image. So what would be ideal would be a faster microscope. So now, most commonly, light sheet microscopes are what are used to image cleared tissue. And the reason for that is we can now, instead of using a point scanning confocal and taking the time to scan a laser back and forth all the way across the sample, we can just send a light sheet in through that tissue, excite an entire nice thin focal plane all at once, snap a picture on the camera, and then move the sample, snap the next picture, so on and so forth. We can move through a tissue really quickly. There's also another advantage to light sheet that if you use a very low magnification objective, something that you generally wouldn't use on a confocal because it would have such a large depth of field and you wouldn't get the Z resolution that you need. We can actually use one of those objectives on a light sheet because now it's the light sheet itself, the thickness of that light sheet, that's actually determining the thickness of our confocal section. So we can throw a 5x magnification objective on there, do slightly larger Z-steps than I was telling you about on the last example. We can now image at 6 frames per second, and we only need 28,000 images now, and it takes about 2 hours to 3 hours to do. So obviously this speeds up the process quite dramatically. So this is what the entire data set looks like once you get a whole cleared mouse brain imaged on a light sheet microscope. So this particular sample is a Thi1 GFP mouse, so it's expressing GFP under the Thi1 promoter in a subset of neurons throughout the brain. So this gives you a good appreciation of the 3D structure of those neurons and how they're arranged within the tissue. Most importantly, this data set, it's about 600 gigabytes in size. Obviously, I've had to downsample it a little bit so that we can make a reasonably sized movie out of it, and I can play it here on the computer for you. So I also want to show you just a small region of this sort of blown up so that you can really appreciate the resolution that's within this data set. So this is a region quite deep into the brain, so I didn't cheat by just using something in the cortex up top. But what you can see is with a nice clear sample with a very strong labeling, which the Thi1 GFP is, we still get excellent resolution even really deep into the brain. So you can still see single fibers, fiber tracts, cell bodies, no problem, even with this low resolution 5x objective, as long as the sample is nice and clear. So quickly, just to finish, I want to give you one example of a project that I think was very interesting and really showed the power of cleared tissue imaging. But before I do that, I'm just going to give you a quick example or tell you a few of the different types of tissue that we've cleared in the facility. So this is a fairly extensive list of what we've done. And I've ordered this specifically. So this is in the order from easiest to clear to hardest to clear. And essentially what makes something harder is the presence of absorbing molecules like heme or myoglobin, or pigmented molecules like melanin, or high fat composition. So basically, the further you go down this list, the more fat there is, the more lipid there is, and as well, the more pigment or the more absorbing the tissue is. So we've done brains, lymph nodes, we've done pancreas, intestine, spleen, heart, kidney, adipose tissue, all to varying degrees of success. Obviously, the brains, like I said, seem to be the easiest thing to do, so we have no problem doing a really big piece of brain. But if you're going to do something like adipose tissue, you're probably not going to be able to clear something on the same level as an entire mouse brain. But you can certainly do it with still substantially large, perhaps a millimeter, a little over a millimeter thick pieces of tissue. Okay, so one quick example to finish up. So this is a paper of the Uchida lab here at Harvard that came out a few years ago now. And the Uchida lab is interested in dopaminergic neurons. And in this particular study, they're interested in finding out which neurons within the brain innervated the dopaminergic neurons within the VTA area of the mouse brain. So they had this very impressive labeling protocol, which I'm not going to go into here, but essentially what happens is after they inject these different viruses, what they're able to do is label with GFP any cell that is innervating a dopaminergic neuron in the VTA. So in the 3D brain that you see down below, that's rotating there, all of those little cell bodies that are lit up, those are neurons that are innervating a dopaminergic neuron in the VTA. So this is just an example of their workflow. Don't pay too much attention to the timeline. Some of this work was done before we had commercial systems to do the clearing, so it took a little bit longer. But essentially what we did was perfuse their animal, hydrogel embedded it, did an electrophoretic tissue clearing process to remove the lipid, placed the sample in a refractive index matching solution, did some Lightsheet microscopy on it, and then started doing data analysis. So in our particular Lightsheet microscope, we'd image half of the brain, rotate it 180 degrees, image it from the other half, and then put those data sets together. In panel C, you can see an example of a single Z-plane from within that mouse brain. And in panel D, you can see all of the individual cell bodies within that particular slice after a segmentation algorithm was applied to it. And then in the graph down below, what they did was they took these brains and registered them onto a brain atlas so that they were able to localize every one of those cell bodies to a particular region of the brain. And this graph below just shows you which regions of the brain, or how many neurons in each region of the brain are innervating those dopaminergic neurons in the VTA. So I think one of the most interesting things about this study, aside from the biology, is that they were able to take almost 100 brains that they cleared. And of those 89 brains, 77 of them were clear enough to image. So not every brain works out perfectly through this process. And this represented a little over 150 terabytes of data. And once they took all that imaging data, did their segmentation and their registration, they came out with this graph at the bottom that's only a few kilobytes in size. So tissue clearing can be a little bit intimidating when you start seeing things like 150 terabytes of data. But this is definitely an example of sort of the upper end, one of the exemplary studies in tissue clearing. And I think what we're seeing, if you look through the literature now, there's all kinds of examples where tissue clearing can help, even on a much smaller level. So for example, just millimeter, 2 millimeter sections of tissue, even trying to go 500 microns into tissue instead of the traditional 50 or 80 microns. That 3D structure can certainly give you useful biological information that you just can't get without it. So finally, I'd just like to thank a few people, specifically Jeff Lichtman, who we've had many discussions over the years about light scatter specifically and tissue clearing in general that have been very informative and educational. There are two postdocs, Joe Bergen and Yo Isagay, and a graduate student, Will Meningas, who originally brought a number of these tissue clearing projects to my desk when I first joined at Harvard, so I probably wouldn't have gotten into this in the first place if it wasn't for them. I'd also like to thank our commercial partners, Logos Biosystems, Carl Zeiss Microscopy, and Arrivas, who have provided us the tools and technologies that we need to be able to complete these experiments and worked with us to make these processes easier and improve our abilities to make them happen. And finally, we obviously couldn't do this without our various sources of funding who have supported some of the projects that I've shown you here today. And of course, thank you to all of you for your attention and for signing in today to listen to this webcast. And I'd be very happy to try and answer any questions you may have. Thank you.
Thank you for your presentation, Doug. It's now time for the live question and answer session. To ask Doug a question, just type it in where it says type your question here and then press submit. So our first question, Doug, and this one asks, what is the best final clearing solution to use?
Yeah, so that's a good question. And there's definitely a lot of options out there. I provided a few of them on one of the earlier slides. The really important thing to think about here is refractive index. In general, the higher the refractive index is, the better your tissue is going to clear up. So it looks like the sort of optimal refractive index for this is up around 1.5. The other thing to think about, though, as well, is that it depends on the optics of the microscope that you're going to use to image. So although you might not get as great a clearing with a solution that's 1.45, if that's what the optics to your microscope are matched to, then that's what's going to provide the best image in the end. So as far as the ones that I outlined earlier, a lot of it comes down to personal preference. So that refractive index is really important. So if you're using one of the homemade ones, you have to be really careful to make sure that you're hitting that refractive index correctly. So you need a refractometer in the lab and be checking these, making sure that there's no evaporation over time from your solution. So perhaps buying a commercial one is an easier way to do it. So Logos has one out there and a few other companies as well. And the only thing that I will say is I have seen a lot of people try to use just 80% glycerol, and that doesn't seem to work quite as well on the clearing side as some of these more specialized ones like the commercial ones or the RIMS, Protos, those types of solutions.
Excellent. Thanks, Doug. And our next question asks, did the lipid removal step also include mitochondrial removal?
No, so it's not removing the mitochondria itself. What it's designed to do is just to remove the lipid which is within the mitochondria. So mitochondria, Golgi, these sort of subcellular organelles that have multiple lipid bilators and they're all folded on top of each other. These are what really contribute to a lot of the scattering in biological tissue. And so what we want to try and do is get all of that lipid out of there that we can. Now, the protein contents of those mitochondria are hopefully, as long as we've done our hydrogel embedding and our cross-linking properly, they're cross-linked to that hydrogel. So the protein components of the mitochondria are going to remain there. It's just the lipid from that mitochondria that's going to be removed.
Great, thanks, Doug. And we've had quite a few questions sort of on a sort of similar subject, so perhaps we can sort of wrap them up sort of within one question. And it's generally, has the clearing been used on other tissue samples, such as plants or lung tissue? And if so, how well has it worked?
Yeah, so it's definitely been used on a wide variety of different tissues. I personally don't have a lot of experience with plant tissue. There are certain techniques within the tissue clearing realm that have been more targeted towards plants. Speaking of clarity specifically, there is one paper out there from 2015 called P Clarity. I believe it was Palmer et al. In 2015 in Scientific Reports. And they showed some clearing using a refined clarity method. They also had to include some enzymatic steps to dissolve some of the plant tissue. But they showed that on tobacco and Arabidopsis leaves. So you can definitely do it on plants as well. Lung tissue, we've done as well. With lung tissue, one of the more difficult things is getting that lung to sort of remain in its native form during your fixation process. So anyone who comes from a lung lab and has done sort of whole mount staining or imaging of lungs will tell you there's some tricks to keeping it inflated while you do your fixation. But yeah, definitely it can be done on a wide range of tissues, and I listed some of those on that one slide, the ones that we've attempted. And as I said, it's just the more lipid content that you have and the more pigment or molecules that absorb a lot of light that are in a specific tissue, the harder it gets. Great.
Thanks, Doug. And our next question is just sort of asking for, I think, sort of more clarification. So with the work that you've presented today, was that carried out on embryonic mouse brain, or was that in sort of within adults?
Yeah, so everything that I showed there was in adult mice, generally 6 to 8 weeks old. The younger they are, the easier it is. Once you get beyond 6 to 8 weeks, there starts to be some autofluorescent deposits that land inside or start to build up within the brain, and those can be really difficult on the imaging side. It's hard to get around those or get rid of that autofluorescence, just because the clarity process itself, its key step is removing lipid. It's not removing autofluorescent proteins or molecules that are in there. So yeah, the sort of 6 to 8 weeks seems to be a really good age to target if you have that ability, and definitely anything earlier than that, embryonic brains are really easy. Definitely helps to go younger.
Excellent. Thanks, Doug. And our next question asks, how does the procedure affect any future immunohistochemistry?
That's a really good question, and I definitely avoided immunohistochemistry there today in the talk. But yes, so this is very variable. Our experience has been every antibody is different, and there's kind of two key steps that affect this. One is your actual fixation steps, and this is just standard immunohistochemistry. Obviously, we all know that our fixation can affect whether our antibody is going to find its epitope or not. So this is just something that you have to test ahead of time with your own antibody. You can do it just with 1 millimeter sections. Try different, usually people using paraformaldehyde, so you can use different concentrations of paraformaldehyde and just see how that's affecting your particular antibody. Another thing that's really important is because we put so much paraformaldehyde into this big, large tissue, if you have any of it remaining that's unbound, or say only one arm of the paraformaldehyde molecule is bound to protein, the other one's just waving around there, it's going to grab your antibodies as they move through. So quenching the paraformaldehyde with 0.3 molar glycine, this is a standard protocol that's been used for years in immunohistochemistry, before you do your immunolabeling is really going to help to mop up that free paraformaldehyde and reduce your background immunostaining. You're going to have to wait a long time. It's going to take a long time for an antibody to diffuse through a tissue. You might have to do your primary and secondary antibodies for 5 to 6 days each if you're doing a really large tissue like an entire organ. Another thing that can affect it as well is temperature. So if you do this at 4 degrees Celsius, you're going to get less nonspecific binding, but obviously it's going to take you even longer. Increasing that temperature may be able to speed up your immunolabeling, but you risk having some more background staining. The other thing is the clarity process itself, the cross-linking of your proteins. Perhaps your paraformaldehyde hits your epitope. Perhaps you get some hydrogel in the way. I haven't found that to be the actual hydrogel itself to really inhibit things, except what I talked about earlier, was if you make that pore size really small, then your antibodies don't diffuse through as fast. And then the last thing, there are some additional solutions out there. So Logos has a new product now that just helps permeabilize the tissue a little bit more and move your antibodies through as well. And those might be an option as well to help on the immunostaining side. But it is very antibody specific from what we found here.
Excellent. Thanks, Doug. Well, I'm afraid that is all we have time for today. If you have asked a question that we haven't had time to answer today, we will try our best to get back to you offline over the coming days. I would now like to thank Doug Richardson for his presentation and for answering your questions. I would also like to thank the webcast sponsors, Logos Biosystems, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you'll join us again soon.